Good morning, students. Today in this video, we are going to talk about mitosis, basically a topic from cell cycle and cell division. This is a topic of 11th science. So let's go ahead and discuss about mitosis. So let's start with mitosis. Here, let's first see the definition. The mitosis is the process of cell division in which one cell gives rise to two genetically identical daughter cells, resulting in cell duplication and reproduction. Now, what does that mean? That means mitosis is the division of somatic cells, division of body cells, when one cell divides and produces two daughter cells, right? And these two daughter cells are same as their parents. They are genetically identical daughter cells, right? Even the number of chromosomes are also same in the daughter cells as their parent cell, and hence this particular type of division is also called equational division. So mitosis is basically a type of cell division where the number of chromosomes remains the same as in parent cell and in daughter cells, and it is also called equational division. Mitosis basically occurs in somatic cells of plants as well as animal. Somatic cells stands for body cells, and it also take place in the germ mother cells. Later on, the meiosis will occur there and to produce the germ cells, but the germ mother cells they have mitosis in them as well. So basically, it occurs in somatic cells of plants and animals. If we talk about a discovery of mitosis, there was a German biologist, Edward Strasburger. So he identified mitosis first in plant cells in the year 1875, and later on there was another German biologist, Walter Fleming. So he identified mitosis in animal cell in the year 1879, and the term mitosis, it was also given by the Walter Fleming in the year 1882. So that was about it's a little bit of a history where we talked about the discovery of mitosis. So let's move on. When we talk about mitosis, okay, here mitosis is basically consists of two main events. So when we talk about mitosis, we talk about two main events. One is called a karyokinesis, where we talk about duplication of nucleus, and a second one is called cytokinesis, where we talk about division of cytoplasm. Right. So first nucleus will divide, and then cytoplasm will divide. So division of nucleus is called karyokinesis, and further it is divided into four stages: prophase, metaphase, anaphase, and telophase. So let's see these entire of phases one by one in the form of a process. So how does this mitosis carry out? So let's just focus on that. So here we come to mechanism of mitosis. So this is the diagrammatic representation of mitosis. We have already studied in cell division that first the cell will divide, and there we have the interphase. There comes the mitotic phase, and then the cytokinesis take place. So this is the entire diagrammatic representation where you can see how the diploid cell is there in interphase, and then it enters into the prophase, which is basically the mitotic phase. Okay, prometaphase, metaphase, anaphase, telophase, and cytokinesis. One by one, we are going to discuss each of them. Here I would like to mention one thing. At times you will get prophase, which is divided into early prophase, middle prophase, and late prophase. Okay, and then metaphase. Or at times we get this prophase as a whole, and then prometaphase as an intermediary stage, and then metaphase. So if we talk about early and late prophase, you need not to mention about prometaphase, right? And if you are mentioning prometaphase, then you need not to talk about late prophase or early prophase. Did you understand? So we are going to go ahead in that way. So let's just see each of these right from prophase to the cytokinesis. Let's see how this mitotic phase is going on. So first thing is a prophase. right? So pro stands for early, before, right? So it is the first phase called prophase, and this prophase is usually the longest and the most complex phase of cell division. So around 50 minutes the cell spends in this particular prophase. So prophase, it can be further divided into three different stages called early prophase, middle prophase, and a late prophase. Here you can also notice the features of early prophase and a late prophase. Okay, so let's just talk about the early prophase first. So here the cell becomes, when we talk about animal cell, it becomes somewhat rounded due to depolymerization of skeletal microtubules. So in the cell, we know that the cytoskeleton is made up of microtubules and microfilaments. So they get depolymerized, and because of that, the structure becomes a little rounded. And if you talk about a plant cell, it undergoes little changes because in plant cell wall is present, right? And here, if you can notice, the organelles like Golgi bodies, like endoplasmic reticulum, they start to break down into smaller vesicles. Gene expression, protein synthesis, secretion, and cell motility, all these activities would be stopped at this particular time. Focus on centrioles here. The centrioles will also duplicate and they will start to move apart from each other. So they will migrate in pairs to opposite end of the cell, and simultaneously they also radiate these smaller microtubules called ester rays. So they release the ester rays. These ester rays, they are in contact with the centrioles, but they are not, they are separated from the cytoplasm, right? So they are in contact with the centrioles. So this is the ester rays. And when we talk about the ester rays, it also helps in formation of spindles, right? So when they move apart, when the centrioles move apart, there the length of the spindles, it increases, and hence they are able to form the spindles, right? So these kind of things basically happens here in case of sister chromatids, in case of spindles. And let's talk about the sister chromatids as well. The chromosomes are visible in the form of thread, right? So the chromosomes are visible in the form of thread. They have started to get condensed. Sister chromatids are also present, and they are attached with each other at the point called centromere, right? So these things are also present when we talk about early prophase. So in the middle prophase, when we talk about the middle prophase, here the nucleoli, it starts to decrease in size and it finally disappears as well, right? Chromosomes will further shorten and it becomes thick. So these events have started. Even the nuclear envelope, it starts to break down. Come to the next stage, which is called a late prophase. As you can see here, how the spindles have been extended from each pole to the equator. Nuclear envelope is completely disintegrated, and spindle microtubules have been formed, and they finally comes in contact with the centromere. In centromere, there are certain points present called kinetochore, and these microtubules, they are basically connect these kinetochore as well, right? So that's how they are connected with the chromosome. So that's all about this particular prophase. Here you're not able to see any organelle, you're not able to see any nuclear envelope, you're not able to see nuclei. Everything get disintegrates. Centrioles will ultimately reach to the opposite pole. They forms the spindle. The spindle will connect, or it will join the chromosomes, right? So these are the activities which is going to occur in prophase. Let's move on to the next one, which is called metaphase. So when we talk about metaphase, it is very short period of time. It occurs for a very short period of time, like 2 to 10 minutes of time. So it takes around 2 to 10 minutes to complete. In this particular phase, the spindle moves and connects with. The spindles are already connected with the kinetochore, and here the chromosomes will start to move towards the equator or towards the metaphase plate, and you can notice how all the chromosomes are arranged on a single line, which is called a metaphase equator. So nuclear envelopes are totally broken down here, and the chromosomes will arrange themselves on the equator. So this is what basically happens, and this is how you can understand metaphase, right? So the chromosomes would be there on the um, equator. Now, once the chromosomes will arrange on the equator, both sides of the spindles, it will pull each other. It will be pulled towards the pole, and hence the chromosomes will break from the center. This phase is basically called anaphase, and that is also very short period of time it takes place, and it lasts only for 2 to 3 minutes of time. So here what happens, that the sister chromatids, they get separated from each other and they start to move towards the opposite pole. Now, the separation of chromatids starts at the kinetochore, and they may form certain, at times, V-shaped structure or L-shaped structure, J-shaped or I-shaped structures, depending on the position of kinetochore. So this is basically happens when we talk about anaphase. So separation of chromosomes will take place in a phase which is called anaphase. And the last phase is telophase, right? So here the chromosomes will reach towards the opposite pole, and then you will see that the chromosome which was the thicker and shorter, it will start to uncoil, and they will again come in the form of a chromatin, thin thread like chromatin. Nuclear membrane will also start to form around these chromatins. So nuclear membrane will start to form, and slowly the other things will also start to form. So nuclear chromosome, which are present, they are actually arranged in the region from the nucleolar material which is dispersed in the prophase. So it will start to synthesize ribosomes, it will start to synthesize the other things which are needed. Okay, the pair of centrioles remain surrounded by centrosphere, and it forms the centrosome now. So these are the things which will basically take place in the telophase. So what basically happens in telophase, that the chromosome will uncoil and it forms long cylindrical thread like chromatin. Nuclear membrane will start to appear, and it will start to form the ribosomes, and the centrioles, they will form the centrosomes. So this is what basically happens in telophase. And the last one is cytokinesis. So that was about mitotic phase, and mitosis, as I told you, that karyokinesis is followed by cytokinesis. So now we have seen how is the nucleus divided or duplicates. Let's see how the cytoplasm division take place. So cytokinesis basically stands for division of cytoplasm. Now when we talk about division of cytoplasm, cytokinesis in animal cell occurs little differently, and in plant cell it occurs a little differently, right? So first focus on the diagrammatic representation of cytokinesis in animal cell. So here what we talk about, that in animal cell, certain proteins like actin and myosin, they participate in the formation of cleavage furrow. So in animal cell there is a formation of cleavage furrow take place. What happens in this cleavage furrow, that certain proteins called actin and myosin, they get arranged on the just inner side of the plasma membrane at one point, and then they start to stretch, and they forms this cleavage furrow, and the cell is divided. So basically in animal cell, cleavage furrow formation take place. But when we talk about a plant cell, here you know that cell wall is present. So there is no cleavage furrow formation take place. Rather, the Golgi bodies and endoplasmic reticulum, they participates in formation of cytokinesis. How do they participate? Here you will notice that the vesicles from Golgi bodies and endoplasmic reticulum, it comes and get fused in the center of the cell. Okay, so by just fusing, they form a cell plate. Then the cell plate, it will extend on both the directions, and finally it divides the cell into two daughter cells. So by the formation of cell plate, cytokinesis take place, whereas by the formation of cleavage furrow in animals, cytokinesis take place. So that's all about mitosis. We have seen how the process of mitosis take place. Let's talk about its significance, right? So here we talk about certain significance. First thing is maintenance of the cell size. So mitosis, it maintains the cell size, right? Once the cell is fully grown, then only it undergoes division, right? So it let the cells to grow, and then only mitotic division will occur. So it restores the surface by volume ratio of the cell, and that's how it maintains the cell cycle. Second thing, it helps in a growth, right? So by repeatedly or by repeated mitotic division, the cell or the zygote, it undergoes the process of growth. So it will form many cells, and then the cells will undergo growth, it will develop, and the fully grown organism can be formed, we can say. So a single cell zygote, it undergoes many mitotic divisions, and that's how the full body can be grown when we talk about mitosis. Third thing is maintenance of chromosome number, or you can say genetic stability. So it maintains the genetic stability. How it maintains the genetic stability? By providing the complete set of genetic information to the daughter cells, right? So it maintains the genetic stability. Third thing, that it also participates in a repair. So the next thing is about repair. Whenever the mitosis, it replaces the old cells and forms the new cells. So that's how it participates in repair. Next thing is about healing and regeneration. So mitosis produces new cells for the healing of wound and for regeneration. So it participates in healing and regeneration. Next thing is about reproduction. How mitosis participates in reproduction? That mitosis brings about multiplication in the acellular organisms, right? Even in case of multicellular organisms and unicellular organisms. So we should not say acellular, we should say unicellular. So mitosis actually brings about multiplication in organisms, and hence it helps in asexual as well as in sexual reproduction. Third thing, that the mitosis, it provides evidence of basic relationship of organism. So you see, uh, all the kind of organism, they have a same way of dividing themselves, right? So most of the organism, you see how they divide themselves by mitosis, and hence it supports the basic relationship of all living things, that we all have something in common, and we share evolutionary relationships. So that is basically the evidence of a basic relationship of organisms. So this is all about mitosis, and at the end I would say thank you.